Hello, guys. We're going to go over chapter 10 here. I'm probably going to put this in all of one video, so this isn't going to be sectioned off into the different sections. But in this chapter, we're going to be talking about cell division in the form of meiosis and talking about how it deals with reproduction, or more specifically, sexual reproduction. So one just little thing before we get into what really the details of meiosis is. What is meiosis? This is when you take a cell here. You can see where we have 46 chromosomes for meiosis, and we're going to divide it twice, and we're going to get 4 haploid cells. Now in here we have 46 chromosomes to start for us, and we're going to get 23 chromosomes in each of these haploid cells. We're basically making sperm and egg in this chapter, or sperm and eggs. One comparison that we can do to mitosis is that the cell is going to start out with 46 chromosomes in mitosis, just like meiosis. However, these cells are going to be diploid, and they're going to have both of the 46 chromosomes in the final cells. Produces 4. Yep, yep, yep. We're good. All right. One concept I do want to review before we move forward is haploid versus diploid. Diploid cells, this is the total number of chromosomes, which consists of 2 sets. You can see here we have these homologous pairs. So these are homologous, these are homologous, and these are homologous. But understand, like, this blue one and this pink one or purple one is not homologous. They're not homologous because they don't have the same genes in the same locations. You can see the centromere differs in each of these as well. Haploid, you only get 1 chromosome per homologous pairing, and the cells at the end of meiosis are haploid. The cells at the end of mitosis are diploid. So what is a homologous chromosome? Kind of like I said before, the homologous chromosomes are the same length, they have the same genes in the same order, and their centromeres are in the same locations. This is like getting 1 chromosome from dad and 1 chromosome from mom. Now, these genes, you're not going to get the same exact gene. So, like, you can get like a blue eye gene from dad and a brown eye gene from mom. Those are called different alleles, and we're going to come back to that in the next chapter, chapter 11. Now, remember that most of the time the chromosomes are not going to be in this condensed form. They're going to look like this. They're just going to be all jumbled about in their chromatin form. That's with an N. You can see here the chromosomes have condensed, but they're not organized, and that's what they do look like at like metaphase or prophase in mitosis and meiosis. However, scientists will organize them into this. This is called a karyotype. You can see there are 1 through 22, and they're organized from longest to shortest. And then we have our sex chromosomes here at the end. There's nothing really special about these sex chromosomes other than they determine the sex of an individual. But we usually put those at the end so we can know what the sex is. And not every species is going to have X and Y chromosomes. There are some that have Z and W and other letters. But for humans, it's X and Y. This is another karyotype. You can see here the red parts and the green parts signify different parts of the chromosome. And we can see we organize them 1 through 22 with our sex chromosomes at the end. Lastly, I just wanted to remind you about the difference between homologous chromosomes and non-homologous. So again, these two are homologous, these two are homologous, these two are homologous. However, like this one here and this one here are not homologous. This one here and this one here are not homologous. They may look the same and they may look like they have the same length, but they are different. They have different genes. Each of these have different genes that do different things within the body. The two X chromosomes technically are homologous. However, the X and the Y chromosomes are not homologous because they have different genes. We're going to talk later on about why the Y chromosome makes a guy be a guy. Now, in meiosis, we have these autosomal chromosomes that we just talked about. Those are the chromosomes 1 through 22. They do a lot of different things in the body, but they don't determine the sex of an individual. You can see here the sex chromosomes. We have an X and a Y and then 2 X's. The X and a Y is a male. The X and the X is a female. Now, the big thing with meiosis is that there are 2 divisions. That's one of the big differences. And we're going to take one diploid cell and to make it 2 haploid cells after meiosis 1. And then meiosis 2, we're just going to further split those chromosomes up. And they're still going to remain haploid. But in this case, we're going to get just 4 haploid cells here with only 1 chromatid per chromosome. And you might be thinking, like, why do we really care about this genetic diversity? Within sexual reproduction, there's a lot of genetic diversity. If you think of asexual reproduction and how bacteria reproduce, there's not that much genetic diversity. And the only genetic diversity they really see is from mutations. Within sexual reproduction, you get a lot more genetic diversity. That's a huge evolutionary advantage. So just a comparison, again, mitosis, there's one division, you create two diploid cells from one diploid cell. Cells are going to be genetically identical, and you want that in your body, you want genetically identical cells. And they're going to create these somatic cells or body cells. However, in meiosis, you get two divisions. This is going to create four haploid cells, and the cells are going to be genetically different. These also create those germline cells, which are like your egg and sperm cells. Now, just like I said with sexual reproduction, this is the fusion of two haploid cells. You can see here the sperm, you can see here the egg, and it's going to fertilize to make the zygote. The gametes are those sperm and egg. If you ever see that word, those are just referring to the haploid cells. And the zygote is that diploid cell that's formed by the fertilization of the egg. You can see here that the egg or the zygote is going to start splitting within that zygote. And you can see the zygote is going to produce something called the blastocyte, and then that blastocyte starts developing into like a I don't know, a baby, if you want to call it that, or a person or an organism, whatever you want to call it. And then, of course, like 9 months later, you're going to have a kid. Oh, I love that picture. I'm going to go back. Remember, you get one of two homologous chromosomes from each parent. So you can see here, this chromosome is here, this chromosome is here, this chromosome is here. This is showing you that half of your chromosomes come from your mom, half come from your dad, but it's specific. You get one of each of these chromosomes that are in a homologous pair from each parent. Just another kind of connection you got to make. We're making sperm cells. I know we've talked about before, but the end product for guys is sperm cells, and this is done in the testes. It's where you make the sperm at. Ladies, you make the sperm in your ovaries. Now, don't get confused by this. You do only really create one egg per first germline cell, but you create these things called polar bodies. They don't actually become like eggs, but this is how you produce one egg. And there are, so you can see, there's still two divisions, and this is still is meiosis, but it's just a little bit different in females. There's a great video here. If you have time, look online for it. It's a fertilization and how the fertilization process works. It's a great video. I'm not going to show here, but it shows you the pathway of a sperm getting to the egg and the fertilization of the egg and how the DNA interacts between the male and the female gametes. There, it's a really good video, but I'm not going to show it here. Going forward, I'm only going to go over the parts of meiosis that differ from mitosis. So essentially, it is very close to 2 rounds of mitosis, but these are going to be the only differences. I could go through each phase: prophase 1, metaphase 1, anaphase 1, telophase 1, but it's going to be really redundant because it's going to be very similar to mitosis. So again, everything that I go over here is just the differences between mitosis and meiosis. So think 2 rounds of mitosis, and here are the differences. First, in prophase 1, we're going to have something called crossing over. Crossing over is the genetic exchange between non-sister chromatids of a bivalent. You might see that word going forward, but it's also, these are just those homologous chromosomes. There's going to be a protein ladder that helps with this crossing over, where we're just going to see flip-flopping of genetic material between the maternal and paternal chromosomes. Now, you might be like, oh my gosh, Mr. Friedhoff, I have to go back and re-listen to that because that was really confusing. Let me show you a diagram of what that looks like, and it's going to make it a lot easier to understand. We can see here we have these homologous chromosomes. That means they have the same genes in the same locations. Let's imagine this is the first chromosome. You got this one from dad, this one from mom. They're going to flip-flop genetic material. They're going to flip-flop this part of the arm. Now, when we split these at the end of meiosis 2, you're going to get one chromatid in each cell. And if you look, each one of these chromatids is genetically different. So what crossing over does in prophase 1 is it creates genetically different haploid cells. Without this, there would still be genetic diversity, but with this, we create a lot more genetic diversity. One misconception that students have when they see this is that they think it happens only once. It happens hundreds of times. I have not found a really great video to show you that this happens all around or all along these two non-sister chromatids, but they happen more than once. We only show you once to make it a little bit easier for you to understand, but then it creates the misconception that it happens only once. No, it happens a bunch. So metaphase 1 is the other place where we're going to see genetic diversity, where we're going to have these homologous chromosomes. They're on the metaphase plate. They're going to independently assort. And I'm going to show you what that looks like with 3 homologous chromosomes, but understand that this is just how the chromosomes assort randomly, and it's going to put the father's and the mother's chromosomes on different sides. So we can see here are 3 homologous chromosomes, and imagine here the blue ones are from dad, the red ones are from mom. So you can see we have this combination, we have this combination, this one, this one, this one, this one, this one, this one. We have 8 combinations for just this or these 3 homologous chromosomes. Now, what that does is you can see here it goes dad, 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 then dad, mom, dad, or mom, dad, dad. And you can see here again, it's just different sequences and different arrangements. And you might not think this is really important, but this is crucially important. It creates a lot of the genetic diversity we see in our haploid cells. Now, for only 3 homologous chromosomes and 8 combinations, you might be like, oh, that's not really that many combinations. But when you add in the fact that humans have 23 pairs, we're going to see a lot of genetic diversity from independent assortment. Anaphase 1 homologous chromosomes are going to be pulled apart. Sister chromatids remain attached. This is different than mitosis. It's a big difference. In mitosis, you rip apart the sister chromatids. Here, they're going to remain attached. This is where it goes from being diploid to haploid because you're going to start out with 46 chromosomes in humans, and you're going to get 23 at the end. Telophase 1 nuclear membrane doesn't reform because we need to split again. Interkinesis is just at a rest period, no DNA replication because they're already replicated. At the beginning of meiosis 2, cells are haploid. So again, we went from diploid to haploid. Cells still contain sister chromatids. Meiosis 2 and mitosis are very similar. Yeah, at the end of meiosis 1, going into mitosis, they're haploid. And then essentially meiosis 2 and mitosis are like exactly the same, except they have half the genetic information. So again, meiosis. It doesn't just have to rely on mutations for genetic diversity. Mutations are good for genetic diversity, but we have two places where we're going to get genetic variations: crossing over and independent assortment. Like I said before, again, with those 23 pairs of chromosomes that humans have, we create over 8 million combinations, not just 8 from where we had 3 homologous pairs, because we have 23 homologous pairs. We're going to create 8 million combinations. Add in the fertilization of somebody else, and you're going to get 70 trillion combinations. And that's not with crossing over. You add in one crossing over event, man, I don't even know what this number is, but it's a very big number. And just understand that creates a lot of genetic diversity. Two people could have thousands of kids, and disregarding twins, they're not going to have the same kids. They're always going to have genetically different kids. The last section here is going to talk about karyotypes and how people may have different numbers of chromosomes. You can see this karyotype is a visual representation of all the chromosomes within a person. We have the 1 through 22 chromosomes, which are autosomal chromosomes, and we have our sex chromosomes, so this person is a male. Now, going forward, I am going to start talking about things like genetic disorders, and it's really important to understand this is just for educational understanding of people and genetics, and it's really important that we respect these people. These people didn't ask for these genetic mutations or these trisomies or monosamies, and it's really important to be very respectful this time. There's a great importance of using the correct terms. And again, I'm not going to say I'm perfect. I've definitely messed up saying what normal is. I try not to use normal because what is normal? No one really knows. That's kind of your own opinion. I try to say the average. So just understand that we're trying to become more respectful of these genetic disorders. And from this point on, like I don't use any memes in class. I don't use any jokes. And I try to get serious because this is a serious topic. Interestingly enough, this is a karyotype of a breast cancer cell. You can see there's a lot more chromosomes than you would expect. You'd expect 46 chromosomes. You can see here, there's a lot more. All right, so nondisjunction is the failure of chromosomes to separate. And what happens here is that you get an increase or a decrease in number of chromosomes. So you either get like 47, 48, or 45, or 44. You can see the euploidy. This is just the average number of chromosomes. Excuse me. For humans, it's 46. For other organisms, it's different numbers. Like for king crab, I think it's like 208. For chimpanzees, it's 48. It's just how many chromosomes that species has. And euploidy is just the change in that number. So, let me go back. We can see here non-disjunction or normal meiotic division here. You can see that we start out with 4 and we get 2 in each of these. However, non-disjunction occurs when a chromosome won't separate correctly. You can see these cells have one more chromosome and one less chromosome. And this is non-disjunction meiosis 2, where we're going to get more and less, and then the average number here. A monosami is when you have a cell which is lacking a chromosome. So for us, it'd be like 45 or even 44. Trisomy is when you have an extra chromosome. And this is, again, when you get an extra chromosome from the non-disjunction. Now, again, going forward, I'm going to show you some images that they're not bad, they're not graphic, but it's just a little bit of warning. Like, hey, these are for educational. There's no making fun of somebody's appearance here. We have to be very respectful. Our first trisomy is the most common, which is trisomy 21. These people are known as having Down syndrome. Again, they have slight mental handicaps, short stature, flat face, heart problems, and some other small characteristics. Uh, they are, again, 1 in 700 live births. Most people have come in contact and interacted with somebody with Down syndrome, and they're some of the greatest people. They're awesome people, but again, they have a trisomy 21, which means they have a third chromosome here. One of the things I talk about in class is as women get older, they have a higher risk of having somebody with Down syndrome. But I try not to scare students and say, hey, make sure you have a kid early. No, no, no. It's just said that you just have to be cautious with this. My mom had me when she was 40, and there is an increased risk, but it's nothing to be really concerned about until after 40, and then your doctor and you will make that decision going forward. You can see here again, as you increase in age after 40, it really dramatically increases. Trisomy 13, this is Plateau syndrome, major circulatory eye and brain defects, 1 in 5,000 live births. Children rarely live more than a few months. I think it's like 90% of kids that are born here die within the first couple months. Now, with the trisomy 13 has, you can see that 13th chromosome there. That's an extra one, which is trisomy. Trisomy 18, Edwards syndrome, 1 in 10,000 live births. Almost all organs are affected. They have a small heart. Again, children rarely live more than a few months. Some students have asked, like, how do people grip with this? How do people, like, what happens here? Some people, and again, I'm not trying to force an opinion on abortion, but some people do choose to have abortions, and it's based on your state and the whenever they find out that the person has trisomy 18. There's a lot that goes into this, and I don't want to go further because it's a tough subject that people have to deal with. But there are, yeah, that's all I'm going to say, and it's a tough decision people have to deal with. Moving forward, though, you can see trisomy 18 has that extra chromosome there. Moving forward, we're going to look at the sex chromosomes and how they can have trisomies or monosamies. You can see here if there's a nondisjunction within the female chromosome or the eggs, you can see we're going to get this triple X syndrome, the X, which is called Turner syndrome, male Klinefelters, and then we don't have this viable because we need that X chromosome initially. Turner syndrome, physical irregularities, short stature, shorter life expectancy, develop as a girl, infertile, wide neck, ovaries don't develop correctly, 1 in 5,000 females. This is where the our person doesn't have a second X chromosome; they only have one. You can see here that X chromosome. Klinefelter syndrome, XXY, usually average academic capabilities, develops as a male that's infertile, 1 in 1,000 babies. 90% of males go undiagnosed at the age of 15. So what that shows you is that most of your genetic disorders will be classified or will be identified very early on in age because that's when you see these symptoms. But after that point, a lot of them will go undiagnosed just because it's not that far from the average male. They'll look pretty similar to an average male. You can see here that XXY. XXX syndrome develops as a female, taller in height, slightly lower IQ, fertile, tend to have average XX or XY and XX offspring. You can see 1 in 4,000 live births. This is one I'm pretty sure, if I remember correctly, that goes undiagnosed. You can see that triple X. I've heard of 4X. I've also heard of penta X, which is like the 5X chromosome disorder. XXY syndrome, or I'm sorry, XYY. This is where a person is called a super Y syndrome, and they're taller, have acne, slight mental handicaps. 88% of cases go undiagnosed, so you can go undiagnosed pretty often in this. And I show a video from VOX, it's Vox YouTube, that talks about the super Y syndrome and how they tried linking it. They tried investigating if it was the criminal gene because essentially, if you have more Y chromosome, you have more testosterone, and that wasn't the case. It was some bad science in the 1960s. You can see here that XYY chromosome. Here's the video. No, we're not going to watch it. So what does make a guy a guy? So with that Y chromosome, the Y chromosome doesn't actually become activated until about 5 or 6 weeks into the development of a person. If a female, with a female, you have 2 X chromosomes. That female is going to inactivate usually one of the 2 X chromosomes. And what's going to happen is this Y chromosome, 5 to 6 weeks in, is going to deactivate the X chromosome. So essentially, this X chromosome is being activated for the first 5 to 6 weeks of guys. and that's why we develop nipples is because only the X chromosome is activated. This Y chromosome is going to deactivate that X chromosome, and it's also going to make things called the SRY gene is going to make things called the testes, which make our hormone testosterone, which makes us a guy. So the reason why we are guys and we have these manly characteristics is because we have this Y chromosome. You can see here this Y chromosome creates that SRY gene, which creates the testes, which creates a bunch of testosterone, which makes us a guy. Females don't have that SRY gene, so they have their ovaries, and then that produces a lot more estrogen. Now, the really, I would say, interesting. I like using interesting in the most educational, respectful way possible. But the most interesting thing here is if you have the Y chromosome somewhere and it's mutated or missing, a person with XY chromosome will develop as a female. So this XY person would normally, I shouldn't say normally, would typically develop as a male, but in this case, they develop as a female. And these go undiagnosed a lot of the time, I should say. Now, this SRY gene can jump and mutate and kind of jump spots and jump locations and jump to this X chromosome. This person, XX, will develop as a male. Crazy, right? It's you know your whole life you've talked and you know that XX is a female and XY is a male, but in these two cases, which again are very rare, but they still happen, they develop as a female or a male, considerably different than what we've maybe learned earlier on in biology classes. So I love this. That gets you to show you that not everything that you've learned, the basics aren't everything. Basic biology is doesn't show you the complexity of life, and it's very different. And again, all of these genetic disorders, I think, really put a spotlight on people are the way that they are because of their genetics. And there is environmental factors, but there's a lot of genetics that go behind why people are the way that they are, and they deserve our respect no matter who they are.